So now we are recording. All right, so let's begin with this question number 1 on this 2016 FRQ, I want to say. Yeah, I think it's 2016. I might be wrong there. But anyway, we're looking at an experimental design that's pretty similar to a design that we've talked about before, which is what we call a choice chamber or choice chamber. And, you know, I don't think Hyewon was there for that class, although I did upload that video. You know, within choice chambers, we are looking at a frequency distribution of where they're going to be, which allows us to sort of predict what we expect to happen if there was no preference and compare that against the data that we have in front of us. So let's just briefly talk about this experimental design, and we'll talk a little bit more about the questions at hand. So it looks like we're working with fruit flies. And as you guys know, we'll see a lot of fruit flies on this particular test because they are the choice model organism for biological research. And I'm just going to draw the fruit fly right here because that's where we're going to put them, right? Insertion point is where we're going to start putting that fruit fly. And what we're going to do is wait about 10 minutes to see if they make a decision. And that decision is whether they're going to go towards the glucose or they're just going to go towards this dry cotton here. And I guess the main purpose of this experiment is to determine whether fruit flies have some sort of a preference towards glucose, which presumably would provide them with a certain amount of sustenance and energy because it's a food, right? It's a food item for them. And in this particular scenario, we can say that we have 3 options for the fruit flies. So the 3 options that the fruit fly can take after 10 minutes is to be in the glucose, or what we call the experimental chamber, or the dry cotton chamber, which would be what we call the control chamber, or they might even be in the middle area without having made up their minds, which means that we actually have a 1/3 and 1/3 in terms of the frequencies at which we establish an expected ratio if their choices would be random. So let me repeat that one more time because that's a really important point. You know, whenever we do an experiment, we're trying to make an assumption that whatever we expect to see could not, could potentially be untrue, as in they may not prefer glucose, nor dry cotton, nor anything else. And what we would expect to see if they don't have a preference is a random distribution of the fruit flies at the 10-minute mark in such a way that they would either be in the glucose, insertion, or dry cotton in about 1/3 of the time for each one of those conditions. Okay, so that's what we're expecting to happen there. Now, before we move on any further though, let's take a look at this experimental design right now and talk about a potential problem, because that problem is going to be part of this B here. It's going to be part of this question in B, but does anyone know a really major problem that we see with the design so far?
Moisture level.
That's right. I think we've probably mentioned this before, but this dry cotton is a big problem. Because remember, whenever we're doing an experiment, we're trying to isolate the driving factor behind some sort of a result that we see. And what we're presuming here in this case is that the driving factor is supposed to be glucose. However, we see that the glucose was actually mixed in with water, and that's why we have a glucose-soaked cotton, whereas on the right side, we have a dry cotton without any water. And that's the problem, because how can we be sure if the fruit flies are more catered towards, not catered, but if they're more, you know, attracted to this side, okay? Not that there are 3 fruit flies at the same time, but if there are more fruit flies going to the left side, we don't know whether it's because of water or because of glucose. So in order to actually isolate for that particular glucose condition, we're going to go right ahead and move on to B really quickly and say, look, one of the experimental control that I could improve on in this case is to add water, right? Is to add water to the cotton. And in that sense, we're trying to isolate for glucose instead of moisture as part of why the fruit flies are moving to one direction or the next. Is there anything else that you guys can think of right now that could be problematic? Even if we did add that water, there's something else that could be fixed. Any ideas there? How about a side preference, right? Right now we have glucose cotton on the left side and we have the dry cotton on the right side. We don't know if the fruit flies just have a natural tendency to veer off to the left or perhaps even veer off to the right. So in that scenario, what I probably want to do is to randomize the positioning of the glucose cotton and the dry cotton to see whether the flies will consistently go towards the glucose rather than the dry cotton, even if it were to be on the right side. So that's probably something that I can do as well. So maybe right here, even though you're only supposed to identify one, another possible condition would be something like left-right randomization to such an extent that we can isolate for that glucose even better. Okay, so those are the two that just kind of comes to my mind right now in terms of experimental variable. There's another one that you could, oh yeah, I just thought of another one. I mean, another one is that this insertion point is a lot smaller than the right side and the left side here. So maybe they just simply don't like being in a small confined space, or maybe they do. So that's something that we could potentially change as well. But again, I think that's going a little bit above and beyond what we need to be looking at. So perhaps the insertion chamber could be expanded to emulate, where they should be. Okay, so those are going to be the experimental control. Now we call this the experimental control because these are the things that we can modify within our control that we can, sort of establish in order to make sure that controls are being held. Whereas the environmental factors is slightly different because when we talk about the environmental factors, these are the things that we could potentially control, like temperature and so forth, but are a little bit different in terms of it being outside of the design of the experiment itself, right? So we're not choosing which side will be glucose. We're not choosing, how much water to add, but rather making sure that the environment in which this experiment takes place is going to be consistent with each of the trials. So what do I mean by that? Well, one thing that you don't want is to have a window over here, let's say. So here's a window. And of course, let's say on the other side is the interior of the room, because what you might expect is more light to come into this side. And, if that had happened, I'm not a Drosophila scientist, but they may prefer that, right? Because I don't think they're nocturnal by any means, but they may not prefer that. And that's certainly something about the environment in which this experiment is taking place that could be controlled. So I would say that temperature light availability is another one. And maybe temperature, as I said, again, this might coincide with the location of, let's say a heater being on this side or something like this. But another one that I think is really fascinating, and you guys always have to think about this too, is the temporality, or the times at which we run this experiment. So for instance, presumably you're going to be running many trials of this experiment. But imagine that you run some trials in the morning and you run some trials in the afternoon and some in the evening time. Well, that's going to be a big problem, especially when we're talking about behaviors of organisms, particularly foraging behavior. Now, foraging means gathering food, right? So in this case, foraging behavior is going to be significantly affected by timing. I mean, if you think about it, nocturnal organisms will forage in the evening time to avoid predators that could get to them in the daytime. And of course, regular daytime organisms might have very specific points of time at which they exhibit that foraging behavior. So for instance, if these fruit flies simply didn't like to eat in the mornings, then those trials are going to show a lot more variance than if you were to actually test them at a time where they actually want food. Now, I'm not saying that this is necessarily true, and many Drosophila scientists will ensure that the timing doesn't matter if they want to run the experiments at various times of the day, but those are some of the factors that you really do want to take into consideration when you're designing an experiment. Does that make sense to you guys for B?
Mm-hmm.
Okay,
Yeah.
great. So that's something that you're just going to have to sort of explain pretty well. And I think, looking over your answer keys, I think most of you guys got that. All right, so now let's go back to A, and then we'll go back to C, because I think C here is going to be the chi-square that we're going to focus on the most here. In A, I think everyone did pretty well. The distribution of the flies in the chamber after 10 minutes, we're assuming that there are going to be more flies in the glucose area. And when you justify this answer, it's rather simple. All you have to say is that glucose is an energy source. And of course, as an energy source, is sought out by organisms. And in this case, we can take it as far as natural selection or evolution by saying, the behavior that allows these flies to sort of orient themselves towards a positive stimulus like glucose being food would increase their survival. And of course, in that sense, that sort of behavior can be adaptive and will evolve over time. Now, you don't have to go that far, but to be completely fair, even if you don't have to know evolution for this particular test, the graders are never going to dog you for talking about something that is legitimate and that actually has explanatory power. And I think for this particular test, which was, I think 2016, but anyway, in this test, evolution was actually covered. And if you could take it to that extent, that would be really great, okay? So A and B, fairly simple. So I think that part was okay for most of you guys. Now, here's where it gets a little bit tricky, because in C, we have a repeat of this experiment with a couple of variations. Now, for one, instead of using glucose soaked cotton and a dry cotton, we're using an end with a ripe banana, which I presume would be a little bit sweeter from my personal experience. And then you have an end with unripe banana, which again, probably not as preferred for the flies, but we'll see how it goes, right? So at time 1, we see that there are 21 in the end with ripe banana, 18 in the middle, and 21 on the end with the unripe banana. And of course, that's sort of the start position for us. And then of course, the end position is that we have 45 fruit flies in the end with the ripe banana, only 3 in the middle, and 12 on the other side with the unripe banana. So what we're going to be asked to do is to do a chi-squared analysis and test for a hypothesis, or in this case, the null hypothesis, that these guys either have or don't have a preference. So I want to start with the null hypothesis, because that sort of gives us the point at which we can sort of begin doing any sort of statistical analysis. So let's do that for a minute here off to the side. So null hypothesis, what is that? Well, let me just tell you, this is really important for you guys to know. Null hypothesis is what you expect to happen if the independent variable has no causal agency on the dependent variable. So to put that simply, it means that the banana, whether it's ripe or unripe, will have no impact on their preference. That's why we call it a null hypothesis. It means that it's nullifying, or it's canceling out the potential hypothesis that something is actually happening. So the null hypothesis is going to be sort of what you expect to happen if your experiment didn't actually work, right? If your experiment had no explanatory power to indicate that the dependent variable, or in this case, the side that they choose, is in any way influenced by the conditions or the treatments of your experiment. So in this case, the null hypothesis would actually be that the flies, I'm just going to write flies, the fruit flies, will show no preference for either, right? For either the unripe or the ripe, okay, or the ripe chamber. That's going to be our null hypothesis. So hopefully you guys sort of know that and should be able to apply that in future studies. So in any sort of experiment that we do, the null hypothesis is going to be the hypothesis that states that there's no effect, all right? And that's what we're going to test. And one of the things that I can say is this: the null hypothesis could be either rejected, and let's talk about that for a second before I move on to the second point. So rejecting the null hypothesis means that something is happening. It means that there is a preference, right? So you reject the null, it means that the fruit flies have a preference. So they have a preference. And if you fail to reject the null, okay, so we don't really say we accept the null. We usually say, the common phrase is that we fail to reject the null. In this case, it really does mean that they do not have a preference. Okay, they do not have preference. All right, so I'm just going to skip out on a lot of these articles like and the, because I don't want to be bogging down your and my time by writing all of that out, but no, they are there. Okay. All right, so let's figure out how we can actually test to see whether we can reject or where we fail to reject the null, and that is going to be done through the chi-squared analysis. And here we have a beautiful chi-squared table already established for us, so we're going to try to see what happens here. So the null hypothesis is as above. So I'm just going to write a little quotations mark here just to show that it's going to be above here over on that side. Okay. All right. So the observed at 10-minute mark, which is what we're looking at, is pretty simple. All we have to do is just transcribe 45 and 3 and 12 from the data that were given to us. However, here's where you guys need to really think a little bit. You see, the thing is, this expected ratio is going to come from your null hypothesis, okay? Because we're testing this null hypothesis, if the null hypothesis is true, what should we expect? That is going to be the expected value. Now, what you'll notice is that if you count all of these fruit flies up, you get 60. And the expected still has to be 60 with a certain level of distribution. So once again, if we go back to this idea that I've written up here, if there are these three options, and if the fruit flies have no preference, then they should simply be distributed evenly across these three different chambers, right? 1/3. So what is 1/3 of 60? Well, 1/3 of 60 is 20. And in fact, that 20 is going to be what we expect if there's absolutely no preference. Because as soon as we say one side is going to be 40 and the other side is going to be 10 and the other side is going to be 10, then what we have is a preference, right? So we want to keep that expected in line with that null hypothesis, in which case we finally have the two parameters that we need to calculate the chi-squared. So the chi-squared formula is actually the sigma of this value right here, and sigma simply means that we add all of this up. So if we do the math, then observed minus expected in the first kan is going to be, or I say kan, the first block is going to be 25 squared divided by 20. This is going to be negative 17 squared divided by 20. And this one's going to be negative 8 squared divided by 20. And if I remember correctly, you guys can do the math here to fill it in yourselves. And that number is going to be something like 48.9, something like that. Okay, that's going to be the number. So far, so good. All right. I want to focus on this formula for a little bit, though. And going back to this sort of reject or fail to reject idea, have a preference versus do not have a preference idea here for a moment. I'm going to write the chi-squared formula again here. It's going to be observed minus expected squared divided by expected. I want to focus on the numerator, guys. Remember, in mathematics, if that numerator becomes really large, then the whole value becomes really large, doesn't it? Right? Okay,
Yes.
so this top numerator is in some ways telling you about the deviation from expected. So how far is the value from what you expect? Because that's what this minus is going to do. It's going to show some level of deviation. So what that tells me is that if the number that you see and the number that you expect, so for example, in this case, when we see 45 and what we expect is 20, that difference of 25 is rather large. And that means that the chi-squared value is going to go bigger, right? Because if you think about it, we're going to have 25 squared, 17 squared, 8 squared. These are all fairly large values. And when those large values get squared, and even if they're divided by the expected, this flat value, in this case of 20, that's going to give us a rather large chi-square value. So what that means is that a large chi-square value is going to be more likely that you are seeing, right, something, more likely to be significant, more likely to be something that is worthwhile investigating, whereas a very low chi-square value is probably telling you that what you see is what you expect. What you see is what you expect, which means that there's nothing significant going on, that they don't have a preference. So a large chi-square value is going to be more likely leading you to rejecting that null, and a low chi-square value is going to be more likely to lead you to failing to reject that null hypothesis. So hopefully, logically speaking, that makes sense to you guys, okay? All right, so we have that chi-square value of 48.9. Now that value doesn't really tell you much. So what I'm going to have to do here is I'm going to have to show you something that we use when assessing that chi-squared distribution. So here's what we need to do with that chi-squared value. Now, if you actually have to use this data to do something on the AP test, they're going to give you this chart, okay? So you don't have to worry about that. Now, what you'll notice is that on the very first column, we have something called the degrees of freedom. And then on the on what we call the row here, the top row, we have all of these 0.99, 0.95. These look like probabilities, or you can multiply this by 100, think about percentages, right? Now, within those tables, within that table, you have a huge array of what we call chi-squared values, which means that the 48.9 is somewhere within this chart. However, where we start is right here on the column where it says degrees of freedom. Now, what a degree of freedom is essentially telling you what row to look at given the number of cases that you have available to you in your experiment. So in our experiment, the number of cases that we have is actually 3, because you could either be on the left side, you could be in the middle, or you can be on the right. So you have 3 cases, but the degree of freedom is always the number of cases minus 1. So in this experiment, because we have 3 cases, our degree of freedom is going to be 2, which is right here. So we have this line that we're supposed to observe here, okay? Now, if you look at the line, you'll see that we start off with 0.02, 0.1, 0.2, 0.5, and so on, which means that the number, the chi-squared value is actually increasing as we go from the left to the right side. In fact, when we go all the way to the right, at the edge of this table, we see a chi-squared value of 9.21. Now, our chi-squared value is actually 48.9, which would be off the chart here, as in it's going to be really, really toward the right side. Now, where are we looking to see whether we reject or we fail to reject the null? Well, that is going to be this column right here that we call 0.05. Now, what that 0.05 represents is sort of a standard with which in biology, or at least in many scientific disciplines, we use as sort of a like a how should I say this? It's sort of this line that you cross, right? So for example, right now we have this 0.05 and we have 5.99 as a chi-square value. And it means that if your chi-square value is greater than this 5.99, that means what you see is significant. What you see is something that is actually deviant of what you expect. And if you want to think about what these probabilities actually mean, it sort of means something like this. The chance of seeing your results, given that the null hypothesis is true, is less than 5%, right? Because it's bigger than 5.99. Your chi-square is bigger than 5.99. In fact, your chi-square is bigger than bigger than 9.21. So if we use the same logic, it means that if you were to run your experiment millions and millions of times, you would only get the results that you see with 45 on the right side and only 12 on the unripe side and only 3 in the middle, less than 1% of the time. Less than 1% of the time. So what that tells you is that, you know what, maybe the null hypothesis is not true. Because if we're seeing this only 1% of the time, given the null is true, that probably means that the null is not true. So we can kind of go back to my tablet here so I can kind of express this data in a more scientific way. What we can say is something like this. We can say, all right, the chi-square value is 48.9, and this represents, first of all, degree of freedom of 2 and a p-value of less than 0.01. Now, it's probably less than 0.001, but this is the data that we have available to us. Now, what does that mean? If the p-value is less than 0.05, now, again, 0.01 is, of course, less than 0.05. So let me just write that here, though, just for your records. Then we reject the null hypothesis. So now let's see the consequence of rejecting the null. Remember, the null hypothesis said the flies will show no preference for either unripe or the ripe chamber. Now, if we reject this, so let's say reject it, what does that mean? That means that flies do show a preference, that the flies do show a preference to one of the two chambers. Now, I can't, without looking at the data, tell you whether they prefer ripe or unripe, because the chi-square, all it does is to tell you that the frequency distribution that you see is different from the expected. But clearly, from the data that you're given, you can kind of make an assumption, or an inference rather, that they do prefer that ripe banana over the unripe banana, and certainly over the middle chamber here. So they definitely prefer this one. And that's how we would go ahead and do that problem. So the null hypothesis we've done. We've entered the values and we've even performed the chi-squared test. So if you're confused about the chi-squared test, I would advise you to slowly watch this video again and maybe run a couple of other scenarios. There are plenty of questions available for you guys on the internet as well. And if you're still confused, send me a message and I can run through this again with you guys in a different, maybe a different setting, maybe a coin toss example, or maybe a genetics example. But you definitely need to know how to do the chi-squared, whether it comes out on this test or not, okay? Any questions on the chi-squared test that we've just done?
No.
No, this is okay, all right?
Mm-hmm.
All right, good. And as I mentioned, I've already uploaded the answer key, all right, for this test on our Google Drive. So, you know, you can always cross-reference that with this video and you'll be able to sort of gain a better understanding of what we're looking at, all right? So that solves C as well as D, so that's fine. Now let's take a look at E. Briefly propose a model that describes how environmental cues affect the behavior of flies in the choice chamber. Oh, that got a little bit cut off there, in the choice chamber. All right, so let me actually tell you a little bit about this. This is a question that you probably wouldn't have seen even if this year's test was normal. And the reason is, what we're looking at here is sort of a question that asks a little bit about physiology, okay? A little bit about physiology. But I will develop this model for you regardless, and I'll do that over on this side where we have a lot of space. So even though they're talking about the environmental cues, I don't think they're talking about light or temperature. I think what they mean here, and they should have been a little bit more clear, their definition of that environmental cue was really the presence of sugar. That's what they meant. And they're saying, how does this as a stimulus affect, okay, how does that as a stimulus affect the behavior of the fruit flies? And in this case, we're talking about, you know, orientation and movement toward that particular stimulus. So let's take a look at what the model would look like. So we can say that on the left side, we have some sort of a cue. And this cue might be, I don't know, let's say a mound of banana, okay? I'm not gonna attempt drawing a banana here. Actually, I can draw a banana. That's pretty, yeah, it's pretty good. And let's say that it's starting to ripe, okay? Now, what we know is that the reason why we can smell things is a result of volatile chemicals that substances are giving off into the environment. So for example, I can sort of draw these smelly lines, but you know, these smelly lines in the real world represents a lot of these tiny little volatile chemicals. Okay, and volatile simply means that it diffuses through the air. Volatile chemicals that are sort of moving away from that source, which would be the ripening banana. Now, here on the right side, let me draw a little fruit fly. Okay. Now, as you guys know, my wife is an entomologist. She could probably draw a lot better fruit fly than I can. But although, you know, I feel like every time I draw, you know, insects, they always end up looking like a bee. I don't know why, because I just have this tendency to draw a big bum on these insects. And that's what those bees and other hymenopterans are known for. Anyway, so here we have a fruit fly, okay? And on the fruit fly, we have this antennae, okay? All right, so let's zoom in on that antennae. And now I'm not saying that this is 100% correct, right? Because again, I'm not a physiologist, but what we can assume is that these antennae are going to have cells, right, that are kind of on this antenna, and they're going to be, you know, involved in reception of information. And if we sort of zoom in on that cell a little bit more, then we can even say that the cells themselves have these receptors on there that are going to be sensitive to certain chemicals that are diffusing through the air. So again, if I were to take one of those volatile chemicals and we attach it to that receptor, then what we should expect is some sort of a signaling cascade that happens, and maybe this is going to set up some sort of a neuron, right, that carries that information, that nervous, right, through the nervous system, that may even end up in the rudimentary brain of the fruit fly that we have here that we call the ganglion. And then of course that ganglion will send messages to the motor units. Of course they have muscles, right? These are animals, and that's going to sort of allow them to orient themselves and move towards that ripe banana. Now, like I said, of all the things that we talked about, really the only thing that you guys really need to kind of understand from this year's test is this segment right there, which is the signal transduction, starting with that receptor, right? Because we know how important that reception is going to be, and we know how important that signaling is going to be. So that's the only thing that you guys probably have to be responsible for. You know, we haven't talked about nerve conduction. We haven't talked about action potentials and motor units and things like that. And you don't need to necessarily for the time being. So we'll just leave the model at that. And I think that is sort of what they were looking for that one question. All right. So what I'm going to do here is I'm going to actually, oops, again, like last time, I'm going to share this with you guys. So you have this on file, and then we can move on to the second question. Oops, that's not you guys. There you guys are. So I'll send it to you guys there. Okay, perfect. All right, great. Any questions on number one?
No.
All right, excellent. That's really good. Okay, now let's move on to number two. I feel like all of you guys fared a lot better on number two than you did on number one, particularly because we don't have any statistical analysis to do here. But this is a really fascinating question, and I really like this question a lot. And there are a couple of things that I want to kind of expand on as we talk about these questions here today. We're looking at what we call the absorption spectrum of two different pigments here. So we can say that this could be mysterious pigment A, and we can say that this is mysterious pigment B. And what you're looking at is the absorption, and of course, by default, we're looking at the reflection as well. So I'm just going to make this black because I don't want to make any, sort of indications that I am actually talking about color, because we are talking about color here. Now, what you know is that on the lower end of the spectrum, this is where the blue and the violets are. And over here on the top end of the wavelength, here we have the reds and the orange and so forth. And in this pigment A, what you'll notice is that this segment right here, from about 500, a little bit less than 500 to about, I don't know, let's say 650 or so, we have a lot of absorption here, a lot of absorption. And remember that when we're talking about pigments, what's being absorbed is going to be what's not seen by your eyes. Because if you think about it, if this area between, I don't know, let's say 480 and 650 is absorbed, what's being reflected is this side here, and also a little bit of this side here as well. Okay, now I say reflected, but I also mean transmitted, as in it's passing through as well. So reflected and transmitted, probably better to say that. And what we'll notice here is that, of course, we have the blue end of the spectrum. Now I'm actually going to use that color here, the blue and the sort of the violet end of the spectrum being reflected, and of course the reds being reflected from this pigment. Of course, when you combine blue and the red, what do you get? You actually get purple. So what we can assume about this pigment A is that it's going to appear purple when we look at this pigment. Now, on the right side, we have the absorbed area that's on the blue end, and we also have absorbed area on the red end. What's being reflected is right here in the middle, and as you guys probably know, that middle is going to be some green color that becomes reflected and transmitted as well. So the pigment B, we can make an assumption that in this case, this pigment is going to show a color of green when we look at it. All right, so now that we have the absorption spectrum figured out, let's go ahead and look at what they're telling us. So what they tell us is that we have one of these spectrums, one of the pigments, which is chlorophyll A, which is found in green plants, and the other pigment is bacteriorhodopsin, found in purple photosynthetic bacteria. So that's awesome, because that tells me right now that this pigment B is certainly going to be chlorophyll A, and then this pigment A is going to be bacteriorhodopsin, bacteriorhodopsin. I don't know if I'm spelling this correctly, but again, the spelling is here for you guys to use. All right, so that part is pretty easy. And I think, in fact, A is actually asking that very question. And in explaining your answer, of course, the answer key is available to you. It's just going to be a repeat of what I said. You know, when a pigment absorbs a color, absorbs a particular wavelength of light, you know, that's being utilized by the plant and not being reflected back. And because we see the colors that are reflected back, you know, you can see that in graph 2, you know, areas of the blue and the reds are absorbed while the greens are reflected and so forth. And as long as you guys don't accidentally mix up your words, I think you guys are all going to be pretty successful in doing that, okay? Now, at this point, I want to pause on moving forward in this question and bring to your attention a couple of things that I think is really important. And it's going to be a little bit of a review. So let me actually go back here and draw this graph that we have for chlorophyll A. I'm going to try to match it to the best of my ability. So something like this, and of course we have something like this. Okay, so this is the chlorophyll A with the wavelength on the x-axis and the relative absorbance on the right side here on the y-axis, okay? All right, now, when we look at this particular graph, and this is the absorption spectrum, okay, absorption spectrum, how did we get this data? How do we actually get a graph that looks like this? What kind of tool would we use? Do you guys know? So, this type of graph can be created by using what we call a spectrophotometer. Okay, spectrophotometer. So what is a spectrophotometer? Well, it's actually a very simple machine. And I remember the first time that I used a spectrophotometer in university, I was sitting there looking at this machine. I remember I was in the university in the early 2000s. I'm not that old, but these spectrophotometers must have been from like 1930s, right? They were super old because of the fact that they're simple. So, I mean, I remember sitting there making a joke with my friends, like, you know, there were like doodles on it saying like Watson was here, you know, or like, you know, so anyway, it's kind of a dumb joke, but yeah, these are very simple machines. So this is what's going to happen. With the spectrophotometer, there's a source of light. So I'm going to say this is light here. And the source of light is going to give off what we call white light, which is the combination of all the wavelengths of light. But this actually goes through a prism. And as you guys know, prism causes different diffraction of light. And what that does is, I'm going to try to do my best to sort of draw out a rainbow here, but you have red and you have orange, right? What's next? Roy G. Biv. So yellow, you have yellow light, and then you have green light, right? And blue light. Let's go ahead and draw blue, and let's draw a pretty dark blue here, violet, like this. So it like sort of spreads that. Oh, that actually looks pretty nice. Okay, anyway, so you get this rainbow. And what you can do is you can actually isolate for one of these lights by saying, okay, well, we have this slit here that I can move up and down. And as I move up and down, I can allow, let's say in this particular example, let's say only green light to pass through that slit. Okay, so I can choose what wavelength that I'm going to be using. And what I have in the middle here is going to be some sample, let's say a test tube containing some solution, right? So in this case, let's say the solution here is essentially water with dissolved chlorophyll. So it's going to be a green solution like this. Okay, just like that. All right. So now at this point, what you should probably notice that, you know, because the green solution here with chlorophyll solution, right? It doesn't absorb green, does it? It either reflects or transmits green. So in this case, a lot of that light is simply going to pass through that particular test tube. So what we can do here is on the other side, have some sort of a detector, right? Have some sort of a detector here that's going to detect how much light is actually being passed through that particular sample. And that can tell you that in this case, maybe we have something like, you know, out of 150 and 0, we have a lot of light that's coming through, telling us that very little is being absorbed, right? Very little is being absorbed. And if we think about the opposite of this, then we can say, all right, well, this is how much is being absorbed. And that's how we create this sort of graph. Now, of course, if this were to be red light instead, so for instance, if I were to sort of have the slit here and have red light, a lot of that red light would be, oh, actually, you know what? This is what I'm going to do. I'm going to try to make it like really artsy here and say, okay, a lot of that red light is going to be absorbed, and only little light's going to come through. And of course, that would mean that you have very little detection at that photo detector, okay, or a photon detector here. Does that make sense to you guys? So that's how we use a spectrophotometer. Okay, so that's the first thing that I want to mention to you guys, because I think, understanding what some of these machines do and what kind of data they spew out is really important. Now, the more important thing is that we usually have absorption spectrum that can be compared to another type of spectrum that we call the action spectrum. So let's draw that action spectrum. All right, so the action spectrum is going to be pretty similar to what you see in the absorption spectrum, but while the x-axis is the same with the wavelength over here, the y-axis is actually very, very different. The y-axis for an action spectrum is going to be the rate of photosynthesis. So we're measuring the rate of photosynthesis. So now, how do we measure that rate of photosynthesis? So that's the million-dollar question. Right? And there's so many different ways to do it, and we've gotten pretty sophisticated at doing this. But the simplest way that many students probably have experienced is by measuring O2 produced, right, in some sort of a system, in some sort of a experimental system. So let's actually draw out how we might get something like this, all right? So here's what we might do. Again, in this case, we're going to use light. But for this light, we're going to put like a cellophane paper. So for example, let's say that we're going to use some sort of a color filter so that the only color of light that's really coming out is going to be, in this case, green. And typically in a school setting, we're going to be using some sort of a test tube like this. And we have a plant that we call Alodia. And Alodia plants are aquatic plants, which is really nice because then they can be sort of submerged into this water. And this is going to be an awful drawing of Alodia, so bear with me here. And what we're going to be seeing is that as photosynthesis occurs, you're going to see the production of air bubbles that come out of the plant leaves. And what are those air bubbles? Well, those air bubbles are actually oxygen gas that is being produced as a result of photosynthesis. Now, again, because we're using green light, we probably are going to be producing fairly low amount of oxygen. Whereas if I were to use red light, and I'm always just kind of comparing red and blue, but if I were to use red light, then you would relatively see a higher level, okay, higher level of oxygen being produced. And that is going to sort of give you this action spectrum. That makes sense to you guys, right? But again, when we're using this action spectrum, we're going to be using what we call a proxy measure. So this is what we call a proxy measure of photosynthesis, because we're not directly measuring the reaction rates that are happening at each of the levels of photosynthetic process, but we're measuring something else that's indicative of what photosynthesis is going to be. Now, of course, in the example that you see in the book, what we call the Engelmann's experiment. So this is the classroom experiment, but in Engelmann's experiment, which was one of the most famous experiments on photosynthesis that we learn about, what he did was he took a filamentous algae. So this sort of a strip of algae like this, okay? I know it looks like DNA, but it's not. Filamentous algae. And then he gave, different wavelengths of light. So some red light there, some orange light here, and yellow light here, and so forth. And what he did was he seeded what we call aerobic bacteria right near the algae. Now, the reason why he did this was, of course, based on the wavelengths of light, right, that spans from here to here, there's going to be differential oxygen production. So you're going to get a lot of O2 here. You're going to get high O2 there. You're going to get high O2 here where the blue light is. You're going to get low O2 production here, right? Low O2 production. And what he saw was that these aerobic bacteria. So bacteria that rely on oxygen doing a lot better near the red and the orange than they did in the greens. And of course, they did pretty well on the blue section as well. So by looking at the sort of growth of the aerobic bacteria, he was able to determine another way to draw up that action spectrum. So that's a pretty ingenious methodology that he used as well for that time frame. All right, so that's fine. But the biggest question is, what's the difference here that AP Bio really wants you to know about? Well, the biggest difference that you guys need to understand is that the absorption spectrum can be done in vitro. And what that means is out of the living in a test tube. Okay, so because we're only looking at the action of a inorganic, well, I shouldn't say inorganic, sorry, action of a non-living chlorophyll molecule, this can be measured simply in a test tube. Whereas here, when we're looking at the rate of photosynthesis, this has to happen in vivo within living systems insofar as they actually need to do the entire process of photosynthesis in order for us to draw up what that rate of photosynthesis is going to be. So anytime we're looking at oxygen production, as Engelman did here, and as you guys may have done in the classroom, that oxygen is being produced as a result of photolysis at photosystem 2. But what we're assuming is that as a result of that, you have the entire ETC working, you have the production of ATP, the NADPH, and presumably the production of glucose through the Calvin cycle. And that's all happening within the context of a living cell. So this is non-living, this is living. And that's the biggest difference that I want you guys to keep in mind. And the reason why I want to stress on this so much is that in so many of the exams that I've seen implemented in the past, some variation of this question came out. And one of the things that they always ask is really whether, the absorption spectrum requires living things or whether the action spectrum can be drawn from non-living systems. So it seems like it's thematic that AP Bio test writers wants you to know that difference, wants you to know what kind of, settings you would be doing this sort of experiment and so on, okay? So that's the first sort of thing that I want you guys to sort of keep in mind. All right, now let's take a look at B. Now this one's not too bad. In this case, we're looking at, look, if we have this chlorophyll A, because they're saying that we're using graph 2. If we have chlorophyll A, and we're looking at a plant, right? We're looking at a plant, and if we give them these different wavelengths of light, and I think they say something like 650, 550, and 430 nanometers. Okay, so these are the wavelengths in nanometers. What kind of growth would we get? What kind of growth would we get, or at least predict the rate of photosynthesis in this case? Okay, rate of photosynthesis. And once again, let's just write O2 produced here, depending on the wavelengths of light. And of course, in this case, going back to this drawing, you'll notice that the 430, which is around here, probably give you the highest rate of photosynthesis there. 550, probably the lowest rate of photosynthesis right there. And of course, the last one, which is 650, you get some rate of photosynthesis, not as high as the 430 though. So, if we do want to compare these, I would probably say something like this. The rate of photosynthesis would be the highest here. That would be the middle of the ground there, and very low in this case. And if we run this numerous times and find the means, then we would probably get some distribution of that data around that mean. Oh, excuse me. Sorry, you'll have to excuse me. What is that? It's April 14th, which is right in the middle of the worst allergy season for me. I'm not allergic to ragweed, but I'm rather allergic to tree pollen. And as you guys look out your window, you'll see the little baby leaves that are starting to come out on the trees. And while that's beautiful, it makes me suffer quite a bit. So I can barely breathe through my nose. Thankfully, none of this is indicative of COVID-19 because those are not indicators of a viral infection. So you guys are safe, double safe because I'm on webcam. But just to let you know, don't worry, I'm not going to be going to the hospital, leaving you guys to do this AP exam on your own. So I am healthy, just too healthy. My immune system is overreacting. All right. So anyway, so I think you guys can sort of justify that as well in this question. All right. Okay. So now my favorite part of this question is C. where it says bacterial rhodopsin has been found in aquatic organisms whose ancestors existed before the ancestor of plants evolved in the same environment. Okay, now it says propose a possible evolutionary history of plants that could have resulted in a predominantly photosynthetic system that uses only some of the colors of the visible light spectrum. So we're going to tell a little story here for a moment because we're looking back into the past. So what I want to do here is draw our oceans. Now, as you guys know, plants are actually terrestrial organisms, and any plants that you see in the water, what you're looking at is kind of like how, mammals went back into the water as dolphins and other, cetaceans. It's kind of like that. We actually evolved on land, and so did the plants. But given that we're going further back in time than plants, we're looking at the ancestor of plants, which are actually believed to be some green algae-like organisms. But before that, hold on, before that, we're going to draw something here because they tell us something. They tell us that in this aquatic system, bacterial rhodopsin came first. So what was here first were these bacterial rhodopsin containing what I can only presume to be prokaryotic organisms that were capable of some photosynthesis. And they're, for some reason, perfectly distributed in that photic zone of the ocean. Now, when I say the photic zone, I mean the area within which light can actually penetrate. Remember, water is not the best in transmitting light. So at some point, water is going to die off. Sorry, light is going to die off, and you're not going to have enough light to perform energy reactions. However, what we've drawn just now is sort of what happened before the algae came around, the green algae came around. So let's talk about this for a moment. See, when you see this purple, what this means is that what they're reflecting or transmitting is really the blue wavelength of light. So there's the blue, because they're not absorbing blue, right? They're absorbing green, blue, and they're also transmitting red as well, just like this. Now, what about the greens? Well, it seems to me that greens are actually getting absorbed. So greens stop right there at these individual bacteria, okay, because they're actually using it. So if you're a brand new organism and you're new on the scene and you want to photosynthesize, well, you certainly don't want to be using green. Why? Because if you decide to use green, you're actually actively competing. You're actively competing against organisms that have bacterial rhodopsin because they're already there, well established. They've already claimed that niche. So what do you want to do? Well, you want to use red and the blue. So then the red and the blue is going to be used quite well by these plants. But by virtue of absorbing red and blue and not absorbing green, what ends up happening is that you become green as you reflect whatever green that does come your way in this photic zone. So if you think about what's happening, it's really about occupying an unoccupied niche, right? That would be a good word here to use, because what we can say is that the fact that the green is being transmitted represents an unoccupied niche for the developing green algae, right? And that sounds funny because they're green algae. They're not yet green algae, but the developing species to say, all right, well, if no one's going to be using this, I'm going to use that. And that's really advantageous because, again, you're not actively competing for the green light. And it almost seems serendipitous that when we look outside, that it's actually green. Because there was a chance, for instance, if this purple bacterial rhodopsin never existed in the first place, there's nothing stopping plants from having been purple or red or whatever other color it could have been. Now, of course, we co-evolve with the plants and have evolved certain reactions to colors, and we find green to be peaceful, and that's fine, but it could have gone another way, which I think is absolutely fascinating in this particular question, okay? So that's how we're gonna deal with question 2, and even though, like I said, the question itself may be simple, it really does provide a platform for us to talk about a lot of important biological concepts. So I really do enjoy this question, and hopefully that makes sense to you guys as well. So I'm going to be exporting this to you guys now, and then I'll take up any questions that you guys might have from this particular question, okay? All right, so now that you guys have that, any problems here from anything that I've discussed so far? Great. I'm gonna take that as no. So here's what we're gonna do. It's already 5:55. Unbelievable, right? Time flies when you're having fun. Well, at least for me, this is fun. What I'm gonna do is I'm gonna pause the recording for now, and then we'll take a short break, 5 to 10 minutes, come back and finish up the rest of the questions. Because as you guys know, the first two questions definitely take the longest period of time. The remainder should be relatively quick. All right, so let's pause now and we'll come back and start again shortly. Does that sound good to you guys?
Yeah.
All right.
Yeah.
So I'll resume recording now. Great. That's going to sound really cool because on the actual video, it's just going to say recording now. Anyway, let me go ahead and share my screen with you guys. So we're moving on to number 3 now. All right, number 3. Now, from this point on, the questions do get a little bit simpler, but you know, I don't make it too simple, right? I want to talk a lot about biology, so I know we're going to be bringing some other concepts into these questions. So let's go ahead and do that. Let me clear this up. You know, I actually use this lecture slides here to teach another class. So there, yeah, I'm a little bit, oh, no, okay. You know what? That's as good as it's gonna get. All right, great. Oh, actually, that's perfect. Okay, so let's go on to number 3. Now, number 3 is not something that I am terribly concerned about for this year's test. However, I mean, it does give us some concepts that I think are important. Unfortunately, it does have some markings on it because, like I said, I use this to teach another class. We're looking at lobe-fin fishes, which are supposed to be ancestor of our amphibians. And they're found in rocks that are at least 380 million years old. And then the oldest amphibian-like vertebrate animal was found in about 363 million years old. So we have these 3 rock samples, one from 350, one from 370, and one from 390. And of course, the answer is going to be 370, given that the sedimentation is going to be representative of how old these organisms are going to be. So for example, you know, our 380 million year old lobe-fin fish is going to be deeper, you know, in terms of geological formation. Is going to be deeper here a million years ago. And then of course, our 363 million year old amphibian structures are going to be at a certain depth here. So there's going to be some meters down here, right? So this is all representative of depth. So we're looking at definitely 370. That's going to give us what we call our transitional form. So this is straight out of evolution. And like I said, evolution is not covered on this test. However, there are a couple of concepts that I think are pretty important for you guys to just know as students of biology. So I want to just mention a couple of things. Now, this example is actually straight out of a real life example or real life thing, event that really was a very famous event in biology. Now, there's a professor by the name of Neil Shubin. Great guy. I had an opportunity to actually go to his lecture when he came to my university. In fact, it was one of my first dates with my wife, right? So she was my girlfriend at the time. And of course, because we're both in the Department of Biology, we're both geeks, we decided to go. And even my buddy from Toronto came down and we went to his talk. And the talk was about his discovery of this exact transitional form. And now we call this animal Tiktaalik. Okay, and the idea behind Tiktaalik is that it represents the linkage between fish and the other tetrapod organisms, tetrapod animals that we see occupying so much of our terrestrial ecosystem. So what is important about Tiktaalik? Well, see, the thing is, for many, many years, scientists had this gap between fish and between tetrapods. And there was really no evidence of, no, not that there was no evidence, of course, there are tons of inferable evidence, but there was no substantial, solid evidence that we could point to and say, look, there's that transitional form. So what Neil Shubin did was that he analyzed these lobe-fin fish, and he also analyzed the oldest amphibians, and he sort of predicted where and when, right? And when I say when, I mean depth, right? Where and when, the depth, he should be looking for this Tiktaalik. And I think it was near Iceland or Greenland. It was one of those two, somewhere up north, and he knew where to go. And I think within a couple of years, they actually found this Tiktaalik. And the Tiktaalik, I would love to show you pictures of them, but I'm sure you could Google that yourself. It really does look like this sort of a halfway point between fish and this sort of a tetrapod organism with these 4 limbs that were starting to develop. And that was really important for a couple of reasons. One is that during this time, evolutionary biology has always been sort of under pressure of, well, certain ideologies, especially from the Christian ideology, stemming from things like creation science or intelligent design. And one of the things that they've always said was how evolution was not a predictive science and it was only retrospective and looking at histories. But this discovery of the tiktaalik really did put evolutionary biology on the map as really being a predictive science, not only predicting where this could be found, but also how far you should dig. That's sort of the thing that you need in order to have a solid science. And I think Neil Shubin did a really great job in sort of putting that on the map, or putting that marker on the evolutionary biology map there. And of course, this was another important feature for understanding how the modern tetrapods came to be, and in understanding what kind of changes accumulated in development of all the tetrapods. So there was a lot of good information that could be had from there. If you guys are interested in this Neil Shubin guy, there's actually a book called Your Inner Fish, which he wrote about this discovery. And of course, this was actually turned into a PBS documentary, which you guys can watch if you don't have time to read as well. All right, so that's really fascinating stuff. Now, this is actually something that I want to focus on more, because I think there is something to be said here that does link to the things that you are responsible for knowing. And it says you want 2 types of evidence that would show that this transitional species did exist between lobe-finned fishes and amphibians. Now, we are looking for intermediate features. That's the key word here, intermediate features. And the intermediate features would really mean that this tiktaalik should possess, let's say, anatomical features. Let's say that that's one. Anatomical features that sort of are between fish and between the amphibians. And this could be rudimentary lungs. This could be limbs that are starting to become more calcified, as in they're starting to become more skeletal, because that's what we see in all tetrapods. Anything like that will work for anatomical features. But the focus that I want to sort of put toward to you guys is really the molecular evidence that we may be able to get. Now, I'm not saying that this molecular evidence would be available given that this is a fossil, but I just want to briefly talk about what type of molecular evidence would indicate a transitional species. So for example, if I say lobe-finned fish here, and let's say I write tiktaalik here, and I write amphibians on this side, we can potentially say that, look, there are these differences that becomes accumulated as these single lineages of organisms evolve over time. And we've talked about molecular clocks before. And if I were to sort of demonstrate what this would look like, and I'm not saying that this is exactly what's going to happen, but if we're comparing nucleotide sequences, and let's suppose that there's something like A, T, T, C, 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 G, C, T, T, A, G, G, C, T, A, for example. And we see the amphibians have something like A, T, T, C, C, G, 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 C, T, T, A, C, 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 T, A. Then we'll note that there are several differences between these 2 organisms, mainly this one right here, this one right here, right? And then maybe this one and this one right here. So there are 4 differences here, right? Now, if we were to be able to sort of analyze the tiktaalik nucleotide sequence, and we see something like ATT, CCC, GG, CTTA, GC, CTA, then we'll notice that the difference here is only this one right there and this one right here. So we have, of course, the ancestral form with the standard, and we have +2 nucleotide substitutions, +4 nucleotide substitutions. Then you can kind of see that from the original, you get 2 mutations, and then you get 4 mutations eventually happening in some gene that is, let's say, neutral with respect to selection. And that would be really darn good evidence that this tiktaalik really did sort of exist between the lobe-finned fish on its way to developing into the amphibian descendant in that scenario. Okay, so that's the type of analysis that I think, to a certain degree, you need to be aware of. because of the fact that you learned molecular genetics and so forth. Does that make sense to you guys, what I've just explained? All right, that's perfect. That's great. All right. Okay, guys, then let's just move on to number 4. So number 4 gives you a very simple schematic here, and they want you to talk about what type of metabolic process, I'm going to put a square around that there, rectangle rather, is happening in this 1 and this process 2. All right, so now here's what I'll say. Anytime AP Biology mentions the word metabolic process, I would argue that 99.9% of the time, they're talking about one of 2 things. 1, which is photosynthesis, and 2, which is cell respiration. Now, we could get a little bit more nuanced, obviously. Cell respiration could be aerobic, as in it involves all of the processes, starting from glycolysis to pyruvate oxidation to ETC to oxidative phosphorylation, or it could be anaerobic, which would involve just glycolysis and fermentation process that's going to recycle those NAD plus molecules, all right, as to keep that glycolysis going for just a bit longer. Now, it turns out that this question is right on the money there. In fact, this top portion right here in 1, I would argue that is photosynthesis, because what we're doing is we're fixing this inorganic carbon dioxide into an organic molecule. Presumably this is glucose. And the second component here is going to be cell respiration, wherein we take that glucose and we rip out those carbon dioxide, or rather we oxidize glucose and we get those carbon dioxides back. All right, so that is the really simple process here. But when they say, you know what, you need to explain how it promotes the movement of carbon through the cycle and talking about the focus of role on energy, I want to just create a brief table here that I think might be helpful to you guys in terms of knowing the right terminologies to use in a question like this. So here's what I'll say. We have 2 main types of reactions that we're looking at here. First of which is called an, oops, I've been doing too many genetic questions here. One of which is called the anabolic reaction, and the other is called the catabolic reaction. So maybe I'm just going to make a table here. So let's say the type of reaction is either anabolic or catabolic. Now, one of the things that we can think about is how that impacts the change in structure of the substrates that we're looking at, like carbon dioxide or sugars. So let's go ahead and write what this is all about. Now remember, anabolic reactions, when we say anabolic reaction, this means that we're creating complexity in structure. Okay, we're creating complexity in structure. So what this means is we take a simple substance like CO2 and we're incorporating that CO2 into a more complex C6H12O2, O2, sorry guys. Now, you know that this is creating greater complexity because the right side of this equation, C6H12O6, is definitely more complex than what you see as CO2, the inorganic CO2, right? Okay, so we're creating complexity. Then what that means is that for catabolic reactions, what we're doing is we're simplifying the actual substrates, in this case from C6H12O6 back down to CO2. Now, I'm not actually adding water here, but you guys get the idea, nor oxygen. All right. So how does this actually affect the change in entropy? Now, entropy, if you don't remember, that's just a level of randomness, or it's a measure of randomness within a system. So when you're creating complexity, you're reducing that randomness. So what you see here is that you have a decrease in entropy with anabolic reactions. And in catabolic reactions, you're increasing entropy because what you're doing here is you're increasing that randomness. So you have more CO2 molecules, it's more randomized in that system. So that's what we're looking at in terms of the delta entropy. Now, when you take all of this together, then for the most part, and I'm not saying this always happens because there are some nuances to this, but there's a change in energy that occurs, all right? And when I say change in energy, what I mean is when we look at an anabolic reaction, when you create complexity and you're decreasing entropy, this is not typically the way that the universe wants to operate. It's kind of like a ball never rolling up a hill, but rather always rolling down a hill. So when you want to put that ball on back of the up on top of the hill, you have to invest some energy. And in this case, we call that an endergonic reaction. It means that the energy is going into the actual substrate from the external environment. And of course, in catabolic reactions, it will be the opposite, which is going to be an endergonic and exergonic reaction. My brain just sort of stopped working for a minute. It's going to be an exergonic reaction, which means that it's going to release energy into the environment. Now, where's that source of energy? Well, or source, or let's say the product that the energy can be stored into. Well, that's going to be sunlight for this endergonic reaction, and it's going to be really just ATP, right? It's going to be ATP that's being created. So ATP is created as a result of endergonic sunlight is used as a result of the endergonic reaction, okay? So that's the table that you kind of want to draw words out of, all right? So that's where the energy is going to come into play. Now, when it comes to the actual carbon, the terms that we want to use is that for this first type of reaction, actually, let me just write it here because that might actually make a little bit more sense. We said that carbon is fixed, okay? Or we can say that carbon dioxide in this case is reduced, as in we add electrons to build those bonds. And of course, on the catabolic side of things, we would say that the carbon, or glucose rather, we could say glucose is oxidized, as in we are stripping away those electrons to break down those carbons off of the larger sugar molecules, all right? And that's what we have for that question. So these are the words that you want to use. Again, I have this answer key available for you guys. So you probably want to see the answer key and see how these words relate to one another, and that might help you. All right, now identify an organism that carries out both processes. Plants. Yeah, that's a good one, because remember, plants are autotrophic, which simply means that they're able to create their own food through photosynthesis. When they break down their food, they're going to be doing the same things that animals do in terms of cell respiration. So plants have both chloroplasts as well as mitochondria. So that would be one simple answer to that B. Okay? All right, great. Any questions on numbers 3 and 4?
No.
Excellent. Okay, guys, then let's keep moving forward on to our number 5. Number 5 is another great question for us to review some really great vocabulary that we need to use. And what you'll notice here is that this is kind of similar to what I've just talked about with those nucleotide substitutions that are used to infer relationships between organisms. Now, in this case, we're not using nucleotide sequences, but rather we're using amino acids. Okay, we're using amino acids. And again, that's not that crazy, because if you remember, those triplet codons that we have from the DNA, right, triplet codons will actually confer some amino acids. So it's another way of looking at it, although we do lose a little bit of the resolution here that could exist if we're looking at the triplet codons. Let me explain why that's the case for just 1 second. Now, if you guys remember, there's a lot of redundancies that are built into the codons, right? Do you remember the codon chart? You guys all have access to the codon chart? I'm going to load one up on my screen now. You guys don't have to load this up, so I'm just going to sort of give you guys a couple of examples here. All right, so let's take a, yeah, here's a good one. So if we have something like CCC, CCG, CCT, and CCA, all of these 4 codons actually encode for an amino acid that we call proline. And this third position is that redundant system that's built in. So even if there is a mutation at this third position here, that's not going to manifest as a change to the amino acid sequence. Now, we usually call that a silent mutation, Now, what that means is when we're comparing different species and we're looking at the amino acid, it may not tell us that there is any difference in the amino acid sequence, given that they all have proline. However, underneath that, you may actually have a nucleotide substitution that could have occurred that would be indicative of some sort of a divergence or a change that accumulated over time. So we're losing a little bit of resolution by looking at amino acids or proteins in our analysis, as opposed to the DNA information. Okay, so that's just something to keep in mind. However, here we're looking at some changes that have occurred between species 1 versus species 2, and species 1 versus species 3, and of course what may have happened in species 4. So that's the point of comparison that we're looking at. Now, let's take a look at 1 versus 2 first. Now, I've already sort of circled this lysine here that has been sort of substituted in place of valine, okay, in place of valine there. So that's a bit of a problem because that is definitely a change that we can recognize. And I don't see anything else. I don't see anything else here. So what I'm going to do here is to sort of give you guys a little bit of a breakdown of what type of mutations exist and what we kind of need to be aware of, all right? So let's take a look. What we're looking at are point mutations. Now, that's actually not clear from this particular question, but I just want to talk about point mutations. So let's just say point mutations. Now, point mutations can be broken down into several types. Now, first, we can say that there is, let's say, a substitution, which means that we have a substitution of one nucleotide with another. Now, we could also have something called insertion, and we could also have something called a deletion. Okay, insertion and deletion. And as the names would suggest, insertion would add a nucleotide where there wasn't one to begin with, and then deletion would remove one where there used to be one. All right. Now, here's the thing. With the substitution, we can actually derive several types of changes that could occur. The first is really something that I've already mentioned. That's going to be called a silent mutation. Now, remember, silent mutation is something like CCC changing to CCG, which then still ends up as proline. So that's going to be fine. However, this substitution can also be, oops, I don't know why that became an eraser, but it could be what we call a missense mutation. Missense mutation. So what is a missense mutation? This is when the substitution of a nucleotide changes the actual amino acid that once used to be in that position. So in this case, we have lysine, we had valine, that became lysine. Now, you know, the funny thing is this missense can still be divided yet into two different categories. See, what we have with valine to lysine, and I don't know if you guys remember this, but valine was a non-polar amino acid that we saw in the sickle cell anemia. All right, so valine is non-polar. But what you'll notice that lysine is actually an acidic, it's a basic amino acid. It's a positively charged R group. So it's an alkaline one. So what you'll notice is that we actually have a change from a non-polar group of amino acids to a basic group of amino acids. So we're changing teams here, and that is going to be sometimes called a non-conservative missense mutation because of the fact that the R groups are substantially different from one another. Now, what's an example of something that would be conservative? Well, that would be something like valine turning into another non-polar amino acid like leucine, for example. Now, leucine is structurally similar. It's a little bit bigger, but it's structurally similar. And because we're still conserving that non-polar amino acid with another non-polar amino acid, we can expect perhaps less of a change to the form of the protein. Now, of course, that may depend on the situation, but generally speaking, as a rule of thumb, conservative missense mutations may not be as bad. All right, so now 1C is going to be what we call a nonsense mutation, and a nonsense mutation is going to take one of these amino acids, so let's say something like valine, and it's going to turn that into a stop codon. So whatever valine codon was, it turns into a stop codon. And what this does is it cuts the polypeptide short. So it cuts the polypeptide short, so it fails to actually produce the entire length of that amino acid chain, but rather stops somewhere in the middle or somewhere in the beginning or somewhere in the end. And as you guys can kind of imagine, where that stop codon comes into play is going to have some effect on the degree to which that polypeptide is going to be influenced. Okay, all right. And in the insertion and deletion side of things, I'm just going to simplify this. You know, when you have a point mutation at a single nucleotide position, you add or delete something, this is inevitably going to end up causing what we call a frameshift mutation. Because remember, this is like skipping a question on an answer key and accidentally writing all the answers wrong, a frameshift mutation is going to cause substantial change. It's going to create a substantial change to that primary structure, and which is going to manifest in that tertiary structure as well. And this is pretty much going to make the protein non-functional, much like what you would imagine with the nonsense mutation as well. Okay, so that's what we have as far as these mutations are concerned. So what we can say is between 1 and 2, we have a non-conservative missense mutation. And in 3, we have a nonsense mutation. And in 4, we have a frameshift mutation. And of course, this would have been caused by a substitution here for 2 and 3. And of course, either an insertion or a deletion here for the last group there. All right, so hopefully that is not too challenging. Do remember those terminologies though. I think that is going to be sort of very useful tools for you guys in answering those questions in that very short period of time that you guys have this year. Now, predict the effects of the mutation on structure and function of the resulting protein in species 4. Well, I've just mentioned down here that a frameshift mutation that we see in that species 4 is going to cause substantial changes. That little delta means change, right? Changes to the primary structure, which may affect the secondary, which will certainly affect the tertiary. Because remember, the tertiary structure is going to be really the R group interactions that we talk about a lot in protein structure. And this is, by the way, one of the main key points of this year's test, because this does sort of connect between the biochemistry unit, the macromolecules, okay, and then all the way to gene expression. So the R group interactions, you certainly want to know what they are. These are going to be from those R groups interacting with each other locally and regionally. We may have hydrogen bonding that forms. We may have ionic bonding that forms. And remember, ionic is always going to be between the basic amino acids plus the acidic amino acids, because they are plus and minus, right, respectively. We may even have covalent bonding forming. And that covalent bonding is going to be primarily formed as a result of cysteine, because they have a thiol group, which is a sulfhydryl group that releases H2 gas and forms what we call a disulfide bridge. So this should be all review for you guys. This is from long time ago, all right, disulfide bridge. We have more, by the way. We also have, I should make this a little bit bigger, but we also have what we call hydrophobic interactions. And that's going to be the idea that hydrophobic R groups are going to want to be on the interior portion of the protein if it's within an aqueous solution. And it's actually going to be on the outside of the protein if it's embedded into the cell membrane. Because remember, the lipid tails in the cell membrane make that region hydrophobic. And then, of course, we have a lot of other intermolecular forces like van der Waals, right? Van der Waals, we talked about that as well. So we have a ton of other components when it comes to R group interactions. So these are the ones that you want to focus on. In the case of this substantial change to the amino acid sequence, we just have to say that the R group interactions are going to be completely different. And as a result, we're going to have a misfolded protein. So let me write that here. We're going to have a misfolded protein. And a misfolded protein is going to have a misshapen, right, misshapen form. And as you guys remember, I always mention this a lot, form and function are intricately tied together in all aspects of science, and especially so in proteins. So when you have that change in form, you're going to have change in function. And in this case, this change in function is probably going to result in the loss of function. So whatever this enzyme or the protein was involved in, it's not going to be able to do that anymore. And as a result, the organism is probably going to have some issues in operating properly. Now, it's true that certain mutations, remember guys, mutations are the primary source of variation in biology. So certain mutations will actually be beneficial. But in the case that we're looking at, we're going to go with the majority of the instances, in which case we're going to get that loss of function, which is going to be bad for that organism. All right. I think I forgot to send you the last image. I'll just do that at the end of class today. But let me send this one to you guys while I remember. Okay. All right. Great. Okay, so that's fine for this question. That's number 5. Just have a few more to go. Let's go with number 6 now. This is a fun question, number 6. In number 6, we're looking at different types of cells, cell type X, cell type Y, and cell type Z. And we're looking at the differential expression, I shouldn't say expression, differential amount of organelles that we have. And from those organelles, we're sort of trying to guess what type of cells they are based on the fact that they are of some eukaryotic organism. Now, they don't all have to be human, okay? And they don't even all have to be animals. Remember, plants are eukaryotic. Protists are also eukaryotic. So we could use sort of a huge array of examples, okay, in sort of predicting where these cells are going to be from. Now, the reason why we do have these different cells, though, is that cells are fairly specialized within multicellular organisms. Remember that, for example, red blood cells are denucleated, as in they don't have a nucleus. They don't undergo mitosis by themselves. They're simply cleaned out by the spleen after their usage is done and recreated in the bone marrow. So, we do get that sort of differentiation and specialization in multicellular organisms. And that's where I might actually begin. Now, first of all, maybe I'll start with Y, because I think that one is the most pertinent to the things that you need to know. Now, in cell line Y, we have a large amount of smooth ER, a large amount of rough ER, a fairly moderate number of mitochondria, but a large amount of Golgi bodies. So what we're sort of trying to do here is by looking at the amount of endoplasmic reticulum and the Golgi bodies, we're trying to figure out, well, what kind of cells would require that much ER, that much Golgi bodies. So then we have to think about the function of those particular organelles. So first and foremost, rough ER. Well, the reason why rough ER is so called the rough endoplasmic reticulum is for the fact that they actually contain a lot of ribosomes that are interacting with that rough ER. So if I were to sort of draw a big cell here like this rough section of the ER has all of these little ribosomes that are attached to it. Now, what are those ribosomes doing? Well, they're actually undergoing the translation process, where they're creating proteins right into the ER, just like this. And that's so that they can vesicularize it and send it through the Golgi and eventually into the vesicles so that they can secrete it out into the extracellular matrix over here, right? Or perhaps they're using it to make some receptors that are actually on the membranes themselves. So I hope you guys remember that the rough ER will result in the production of proteins that are either secreted or embedded into the endomembrane system. And what about the smooth ER? Well, the smooth ER here is going to be the lipid production center. So if we have a lot of vesicles that are required for these movements, the trafficking, then you're going to need a lot of that too. So I would argue that this Y cell here is going to be a cell that's involved in secretion of things, so secreting cell. So what might those cells be? Well, I know we haven't done physiology, but those might be something like a pancreatic cell, any cell that's involved in the endocrine function or the hormonal function. As you guys probably know, pancreatic cells, specifically the ones from the beta cells of the islets of Langerhans, are involved in things like insulin production, glucagon production, and so forth. These are, hormones that are supposed to be released out of the cell so that they can sort of enter this blood vessel that are adjacent to those cells so that they can carry that message to the liver and to the vessels and all that. So in this case, we're going to be looking at a secreting cell. Now, the next one that seems pretty easy is Z here, which, by the way, is missing all of these different organelles. So the first thing that comes to my mind is that this cell is really non-functional in terms of the internal mechanisms that a cell would need to do. So what kind of cells would be non-functional with respect to its internal mechanisms? Structural cells, perhaps. Oops, sorry guys, I'm just going to simply cancel this share here for a second because I am getting a little bit of a delay here. I think it might be the Wi-Fi connection, so let me just connect that again. So hopefully we'll have a little bit of a smoother picture here. There we go. Yeah, it's a little bit delayed. I think my Wi-Fi is slowing down. I'm just going to go ahead and write out what I need to write out, and hopefully this will catch up. Let me try that again. Guys, is my audio and my webcam feed coming in okay for you guys right now? It's not too delayed?
Okay. It's okay? All right, so it must be this sort of connection between the program and my iPad. So I'm having a little bit of trouble there. So let me try this because it's not working right now. So, all right, just give me 1 sec, guys, no problem. I mean, we're almost done here. So what I can just do is to bring up these this test on my computer. Let me actually just search for it. This might be faster. By the way, this is 2013. Okay, so I said 2016. I was wrong. It was 2013. So that's just as well. All right, this is what I'm going to do. I'm going to share my Google Chrome. This works, right? Now, unfortunately, I can't write on this, but we don't have a ton more to do. So let me just zoom in on what we're looking at here and say, all right, well, we got these cells that are going to be non-functional, most likely structural. So I have a couple of ideas. One is that in plants, we have these cells called the xylem. You know, there are advantages to simply using this as well. So if you look at this, you'll notice that xylem is responsible for the transportation of water. And the reason why they've drawn this as brown is that the xylem cells, or these vessels that we create, are dead at maturity. So when they're actually functioning as these vascular elements, they're actually dead cells. So that's a really good example of what that cell Z might be. Alternatively, you could have said skin cells on animals, and of course the skin cells, the epidermal cells, the outermost epidermal cells, would be simply dead skin cells anyway, so that would also be indicative of all of these organelles being absent. Now the trickiest one is cell type X, where we have a large amount of mitochondria and the presentation of cilia, and of course small amount of everything else. Now here's a couple of things that you need to keep in mind. From the matrix of information that you're given here, you'll notice that this cell is the only one with cilia. So you have to talk about that. You have to incorporate that somehow. And the way that I would do that is by sort of linking this to the large number of mitochondria that I have. Remember that mitochondria is responsible for the production of ATP. Now remember cilia, which are these little hair-like projections from the cell, right? Every time they move, they're going to require ATP. So both of these guys are working in concert of one another by mitochondria producing a large amount of ATP, able to move that cilia very efficiently. Now, what might this cell look like? Well, this cell could be, let's say, a protist that has these cilia so they can move around efficiently on the outside. So that'll be a single-celled organism example. Alternatively, you could say that these are sort of cells that are involved in transport of materials outside of the cell. So by the movement of cilia, it might be creating some sort of a current in the extracellular fluid so that it can move things around. That would be okay too. One example that came into my mind, and it's not exact, is that sponge cells actually have, that sort of function. If you look inside, remember that these sponges are filter feeders, as in they need to make sure that the water is flowing. And in this case, we're not looking at cilia, we're looking at flagella, right? So it's not going to be exactly what you can say for this particular answer. Although if you said it, I don't think you get it wrong. But these would be involved in the movement or creating some sort of a circulation pattern as these flagella would flap back and forth, or actually flagella would rotate, but cilia would move back and forth. But you get the idea. So these sort of current forming cells may be what the cell type X is going to be. Okay, so you can sort of use the data to talk about that. All right, so I know that that question was a little bit tricky, but you guys should have been okay, given that you give it a little bit of thought with respect to that table. I'm going to try to share my screen one more time, see if it's a little bit faster. Yes, we're back on track, so we're okay now. So let's move on to the next question. I don't think I need to necessarily send you guys this particular image, so let's move on to question number 7. All right, so question number 7 is fairly simple. Again, this is a little bit of physiology, so I'm not going to kind of dwell on this question too much. However, I do want to say that, from the experimental perspective, the one scientific question that we can ask, and I know you guys did all really well here, so I'm going to go through this very quickly, is what is the effect of ethyl alcohol on urine formation or urine output? So just to give you a little bit of background as to what we're looking at, we're saying that the ethyl alcohol is a substance that we call a diuretic. Diuretic is a substance that increases urine production. And in some other examples, we might say coffee is a diuretic, or caffeine for that matter. Alcohol is a diuretic, and so on, all right? And there are some other diuretics too that, you know, is just sort of escaping my mind right now at the moment. So this diuretic is clearly working because, as you can see, at 1% ethyl alcohol, we have that increase in the average urine output. 5%, we have yet another increase. These are trends, okay? So without some sort of a statistical test, I can't actually tell you whether this is significant or not, but there definitely is a trend in increased urine output as a result of ethyl alcohol being added to their fluid ingestion. The hypothesis that could be tested would be that increase in ethyl alcohol in fluid ingested would result in an increased urine output. And this hypothesis could be tested using a linear regression. So for example, right now we have, let me draw that a little bit straighter. We have a situation where we have water, we have 1%, we have 5%. Now you could imagine that we can sort of continue onwards and can say, all right, well, average output is 3.5, 3.8, and 4.7. And we might be able to sort of get this model of how ethyl alcohol may affect this particular rat's urine production, or the species urine production. We'll get some sort of a R-squared value, and we would also get a p-value as well, hopefully less than 0.05, to indicate that there is, in fact, a significant correlation. So that's something to think about when you guys are looking at any investigations in science. So the effect of alcohol on urine production, we just have to say that there's a trend of increasing urine production as a result of increase in the ethyl alcohol consumption. If you want to mention that there's insufficient data to state that the data is significant or not, that would be really great. That would be really great. And the reason why you don't have insufficient data, reason why you have insufficient data, is that we're missing what's happening around the mean here. So remember, we talked about how the difference of means could only be analyzed properly so long as those differences are greater between the groups rather than within the groups. So we don't have that data, so we can't really say for sure whether it's significant or not. Now, I don't think that's what they were necessarily looking for, but hey, if you can give them that, I think they're going to be very happy. All right? Okay. So we're finally at the last question, guys. Wow. So today's been there's a lot of information today, so hopefully it's been helpful to you guys. So let's finish on a high note here. In this case, we're talking about signal transduction. Now, this is from Cell Communications, I think chapter 11, if I'm not mistaken. We focused a lot on this chapter because I really think this is one of the most important chapters for this test. As I mentioned, signal transduction pathways really do connect a lot of different concepts in biology together, things like ligand and receptor interactions to the idea of kinases and how those enzymes operate, and of course gene expression that could be moderated or modulated by different pathways. So I think it's really important to really get a good understanding of the mechanisms involved in cell signal transduction. And they want to talk about these 3 steps. Now remember, the first person to really outline these 3 steps was a scientist named Sutherland, E.W. Sutherland, and he was looking at, it was actually epinephrine. He was looking at epinephrine and its effect on glycogen phosphorylase and the production of glucose from glycogen in the fight or flight response. Now, he said that the first step is going to be labeled as reception. Okay, and we'll go into details about each one of these as we go through this question. The second was the transduction of that signal. And then the last part here in step 3 was what we call the response, okay, the cell's response. Now, this response is pretty varied depending on what type of cascade pathways you're looking at. However, step 1 and 2, we saw that there were some similarities that were happening within different systems. Okay, so let's talk about it for a moment. The importance of course, the reception is that the signaling molecule, okay, so this is the signaling molecule. I think that's 1L, but excuse my spelling there. That signaling molecule is going to be
attaching, in this case, to what we call a membrane-bound receptor here. And this is really the doorbell, right? Because without this process, this cell has no idea that someone is trying to talk to it, right? That's the idea. But the thing that I want you guys to be really aware of is that this relationship between the ligand and the receptor is going to be a result of course, again, that proteins form, the active site right here, how that amino acid sequence or the residues of amino acids are going to interact with this particular ligand, and this could be a protein, but it doesn't have to be, how those chemical reactions interact with one another, which is going to trigger a conformational change on the inside of that protein. And usually that change is going to activate some pathway that's going to happen during the transduction process. But it's really the change in the chemical interactions here that's going to change the chemical interactions downwards. That's going to have an impact on changing that conformation, maybe a little bit of a residue sticking out there that's going to allow some other chemical reaction to take place. Now, when we talk about this reception, we had three types of receptors that we learned about. We had G protein coupled receptors, which would be sort of linked to this G protein here. And again, in this case, this conformational change would replace that GDP with GTP, activating that G protein that can go ahead and interact with other enzymes and turn those enzymes on. Another one that we saw was something called receptor tyrosine kinases, which existed as these little single receptors that dimerize as a result of ligand binding, and then that would act as a kinase, as in it would be able to take those phosphates from ATPs and move those phosphates onto subsequent enzymes in the pathway. And we also had, gated channels, and these might have been, ion-gated channels. We generally call them ligand-gated channels, but they could be ion-gated, or they could be things like IP3-gated calcium channels in the endoplasmic reticulum. Those are some of the examples that we saw in class. So that covers the reception portion. Now, in the transduction process, I want to say that you guys probably want to mention two things here that are important. And the first one is that the cascade acts as an amplification system. Okay, it acts as an amplification system, and that's because one kinase can activate many kinases, usually 10 or more. And each one of those kinases can activate, more kinases. And the more complex system that we have, the greater amplifications that we may see. And that is really important because we want to get a lot of things done very quickly as a result of this signal. So amplification is certainly going to be one of the main things that you want to mention here. And another one is the crosstalk that could happen between several pathways within the cell. Now, again, we're looking at a very simple model, but if you look up any signal transduction pathway that's worthwhile investigating in today's world, you'll notice that the arrows are just all over the place because there's so much going on. And that crosstalk really allows for the fine tuning of a response to the extent that we're looking at, concentration battles, really, the concentration of certain kinases versus concentration of phosphatases, concentration of X versus concentration of Y. That's going to take this sort of an on or off switch and give you an amazing array of response that could be continuous in nature. So it's not discrete, but rather continuous in terms of its severity, okay? Now, this response here, as you guys can tell, is not a simple cytoplasmic response, as in the final is not some enzyme being activated to turn substance A into substance B. In fact, what we have here is a response that links to what we call gene expression. So typically, when we look at a model of gene expression being modulated by a signal transduction, this final activation here, that final substrate of activation is most likely going to be some sort of a transcription factor. And as you guys remember, transcription factors are important in their relationship with the promoter regions of the gene. So for example, if I have a promoter upstream of a gene that I want to transcribe, all right, so here's the transcribed region, transcription unit, right? Unit here is going to be upstream of that transcription unit. And it usually has a Tata box involved as well. And that means that, it's
this transcription factor that comes in, can mobilize other transcription factors, and that eventually allows the RNA polymerase to land or bind so that it can go ahead and make those pre-mRNA. Now, why am I saying pre-mRNA here? Well, because I see the nucleus, which means that this certainly is a eukaryotic organism, which means that you have to process that mRNA in order to get your final messenger RNA back out into the cytoplasm. So there's, that's just going above and beyond what we need to mention here. So you don't have to go that far out, but the point is that this transcription factor being activated, usually through phosphorylation, is going to allow certain genes to be activated. Okay, so going back to the signaling molecule, this is a hydrophilic signaling molecule. This is aqueous because remember, if this were actually hydrophobic, like for example, a steroid hormone like testosterone or estrogen, it would actually go straight through the membrane and go and bind to an intracellular receptor. All right, and I know that testosterone and estrogen are well known for their regulation of gene expression. But what's a hydrophilic ligand here, signaling molecule that may be involved in gene expression? Maybe it could be something like a growth hormone, because a growth hormone is in fact water soluble, and they do bind to a membrane-bound receptor, but they yet activate a transcription factor. So a growth hormone may be what you're looking at there. Okay, so that gets us through the last question. How's that for you guys? Any problems there?
Nope.
Good. Now, what I want to mention here is that none of the things that I've mentioned today should be new. Many of the things that we've talked about ought to be fairly fresh on your mind and is definitely in the textbook. All right, and you guys all have that Campbell biology textbook. So if there's anything from what I've talked about today that you guys are a little bit unsure about, that you guys feel not as confident about using the proper vocabulary or even explaining the proper context, I would highly advise you guys to go back to your book, find that chapter and read it. I know it's a college level textbook. It's not that easy. However, I know that you guys are capable of making notes and simplifying things and breaking things down. And one of the best ways for you guys to really get this into your knowledge permanently is to make models yourself, is to draw things out. And when you draw things out, what it allows you to do is to connect conceptual ideas together. And remember, this AP Biology FRQ is not a regurgitation test. It's not about, spewing out vocabulary and just answering questions after questions. This is about connecting the dots. So you really want to make sure that you have a good idea of what's happening. And one of the best ways to do that is to simply keep drawing conceptual models until you're fully capable of explaining that in your own words. And that is going to be critical in successfully writing this exam. Now, I'm going to stop recording at this point because we're done with our lecture.